Hi guys, welcome back to the Learnify channel. Today we're going to be looking at cell cycle regulation. What cell cycle regulation is the process of many checkpoints being part of the cell cycle, specifically G1 to S and S to G2, where cell cycle division or cell cycle progression can be halted if there are any errors. So there are two main ones you need to know about, which is G1 to S and S to G2. And there are four main types of molecules. The molecules that are important here are called cyclins and CDKs. And there are four types of cyclins: D, E, A, B, and that's the order in which they're used. And we call them dogs eat apples and bananas as a mnemonic to help us remember. And CDKs stand for cyclin-dependent protein kinases, which are a type of proteins where cyclins bind to them. And these activated proteins can add phosphate groups to other molecules in order to help cell cycle progression. Anyways, there are two other types of molecules you need to know about. These are Rb and P53, which are both molecules that inhibit the progression of the cell cycle. And why is this important? Because they check for errors in cell cycle progression and prevent the cell from, of course, dividing and replicating, because we don't want error cells or mutated cells to progress. So Rb and a P53, they stop the cell cycle. So let me draw a stop sign here to illustrate that. And what can happen is these can be inactivated. So if we inactivate something that's supposed to stop the cell cycle progression, we can actually continue the cell cycle progression. So Rb can be phosphorylated, right? So like CDKs, they can phosphorylate the Rb, and this allows DNA replication to occur, and so the S phase can progress. So in terms of cell cycle, if you may recall, it's G1 to S to G2 to the M phase or division, and S phase is where we have DNA replication. G1 is growth of organelles, increase in size. G2 is more like preparing for mitotic spindle and also growth than the M phase. So in that sense, we can call Rb a G1 checkpoint that prevents the cell from going to S phase. Now, P53 acts as an inhibitor by inactivating CDKs. So CDKs, cyclin-dependent kinases, are important in cell cycle progression. What P53 does is sort of inactivates them, and this again helps to hint that this is important in cell cycle progression from G1 to S phase for Rb and P53. Now, what can happen is these Rb and P53 genes can actually be mutated, and that's a common part of cancer, because if you have mutation in these genes, which are really important for cell cycle regulation, you can have uncontrolled division of the cells, leading to cancer. Okay, now moving on to G1 regulation, we have cyclin D, again the first one, plus CDK4, which cyclins bind to CDKs, and this leads to the formation of cyclin E. Now, cyclin E, that binds to something called CDK2, and this in turn prepares the cell for the S phase. Now, it all looks like scribbles here, the notes, but I'm doing that so you guys take your own notes, and that will be more helpful than just looking at my video. How exactly does cyclin E plus CDK complex actually promote S phase? It actually increases the transcription factors, trans factors, and other molecules that assist in replication. So it's just like a bonus. It keeps adding to the replicability of DNA and DNA replication S phase. And in the S phase, the active CDKs can actually allow the formation of the pre-replication complexes, which are important in DNA replication. So, for example, single-stranded binding proteins, DNA helicase, all those structures being taken together in order to have the pre-replication complexes formed, so DNA replication can occur faster. So in terms of cyclin E and CDK2, we actually have the formation of something called cyclin A. And this is a bit more specific, but you won't really need to know that cyclin A binds to CDK2, and that forms cyclin B. Now, cyclin B is the last one in this step because dogs eat apples and bananas. Cyclin B is to do with mitotic spindle formation, so the M phase, so from G2 to the M phase. And cyclin B binds with CDK1. So as long as you guys remember this formula, so for G1, G1 checkpoint cyclin D plus CDK4, and once you're done with G1, you're to move to G2 phase. For the S phase, you need cyclin E plus CDK2. Then you need, when you're in the S phase, you need cyclin A plus CDK2 to go into the mitotic phase, to go to G2 phase, where then you need to move to the mitotic phase through cyclin B and CDK1. Just remember the pairings of the CDKs and the cyclin, and you'll be good to go. And that's a cell cycle regulation in under 5 minutes. So make sure you guys smash the like button, leave a comment down below with any suggestions, and subscribe to the channel. Have a good one.